Okay, now that you're back from the pause, I hope what you've observed here is that if I take histidine, which is a basic amino acid, and aspartate, or aspartic acid, this is the name, by the way, for the conjugate base of it, just like acetic acid. If I take the H plus off, I get the acetate ion. Same deal here. You'll see aspartic acid sometimes called aspartate, glutamic acid sometimes called glutamate. I do it without thinking about it. I apologize. I'm trying not to, but I want you to be prepared for the real world. Histidine and aspartate, basic amino acid, acidic amino acid. This one's going to be negative because it's lost its H+. This one's going to be positive because it's picked up an H+. This is going to give you a salt bridge. Our next one here, alanine and valine. Recall that alanine, side chain is just simply a CH3. Valine, we've seen a couple of times now. Its side chain is an isopropyl group, all alkane all the time. These guys are going to give you nonpolar interactions and probably end up in the center of your protein. Two cysteines we just talked about. Those are the ones with the thiols that we see right there. Two of those able to form a, a disulfide linkage. Serine and asparagine, if I look at these two, remember asparagine has a nitrogen, but don't get distracted. It's not an amine, it's an amide. So that is not a basic N. This is not positively charged. It is, however, polar. And so that will allow it to interact favorably as well with the serine. And so these two would be able to interact with each other. They'd also pretty favorably interact with water. You'd expect to see them on the outside of your protein, okay? Now, last but not least, sometimes after you've done all of this good stuff, with a globular protein, with a single protein chain, you might still have hydrophobic areas, or unpaired ions, or unreacted thiols, anything that could potentially interact with something else, with some other amino acid on the outer surface. When you have that, you might actually only be able to satisfy these proteins and keep them dissolved if they interact with another protein. Or if you're talking about unpaired ions, it might just simply be that they would very much attract another protein that had the opposite charge, or that had another cysteine to react with an unreacted thiol. When this happens, when I take those same attractions, although, like I'm saying, mostly the ones that aren't polar interactions tend to be involved in this, when I take those interactions and use them to put several different protein chains together, what I've got is what we call quaternary structure. And a very important protein in your body actually uses quaternary structure, and that's hemoglobin. Hemoglobin is built from 4 protein chains. Two of them, there are 2 pairs of what are called identical subunits. So there are 2 of these alpha subunits, 2 of these beta subunits, and it's only when all 4 of them come together by virtue of some thiol reactions, some disulfide linkages being formed, and a lot of hydrophobic interactions, a lot of nonpolar interactions. It's only when they all come together that they're able to stabilize in this little prosthetic group called a heme that contains the iron atom that bonds oxygen and that keeps, number 1, oxygen in your blood, and that is also responsible for why your blood is red. It's all about that iron that's been oxidized there at the center and is bound up to the heme. That only works out when the whole thing is assembled this way. So just to get a sense of how you're doing with the different levels of structure, I've attached a couple of questions here, or rather a couple of statements that I'd like you to try to define as being either about primary, secondary, tertiary, or quaternary structure. And I'd like you to really focus on finding some words that help out with this. So I think you've got all 3 of them there. As I'm doing this, can't remember whether it's going to box in the first one if I hit the next down. So I'm going to ask you to look at the worksheet and try those out, and I'll find out in a moment. Pause the video, come on back, and we'll talk these out. Okay, let's see. So isocitrate dehydrogenase is an enzyme that is made from 8 identical polypeptides. Let's get your answer here for it. What did you think when you saw this that it was talking about? Primary, secondary, tertiary, or quaternary? Okay. I hope that if you focused in on the 8 identical polypeptides, all having to come together, that you chose quaternary structure for that one. The next one, enzyme bovine ribonuclease, contains 5 molecules of aspartic acid, all of which are on the exterior of the protein. So let's think that one through for a moment. You're being told about one of the amino acids. Aspartic acid, if we think back to it, is a acidic amino acid. That means it's going to have a negative charge on it under physiological conditions because it loses its H+. And we're talking about where it winds up. And if I'm looking at something that is charged and it ends up on the outside of the protein, we're talking tertiary at this point. Particularly in this case, we'd be discussing polar interactions. If I went to tell you about how aspartic acid interacted with a histidine or with a lysine, where it was an acidic and basic, you might then tell me that it was an evidence of a salt bridge. That would still be tertiary structure, though. It's all about where the individual amino acids end up in the final folded structure. Lastly, we've got bovine ribonuclease contains 3 sections of alpha helix. Well, this one I hope is not too hard. If I've got a bunch of alpha helix, all I'm really describing is the secondary structure. I haven't talked about where those helices end up. I haven't talked about the side chains on them. I'm just saying that it's got this particular very common structure, okay? Now, when we come back for our next video, you're going to take a look at how we break one of these structures, how we take these forces and adjust the surroundings to allow them not to do what they want to do. And some of this you might recognize from some of the battle against coronavirus. So come on back and watch that one at your leisure.